Mm.
Okay, so I haven't done that. Okay, let us see the requirements of the vectors. Like what are the things that are essential for a vector to be a vector? So first thing is origin of replication. Now you, first of all, you see, like match it to your notebook. These parts you've written, DNA ligase, DNA polymerase.
Ma'am, can I just take a notebook and come?
Yes, please.
I, or I can see it later because my writing I have left it like before, like way before.
Way before all of that. But I
Yeah.
believe that I have
But
taught
we
you this.
finished. We thought you taught me, but I didn't write it completely. That's the thing.
See, because I remember Malayalam,
Ah,
mom, dad.
this I didn't write yet. This we finished doing, but I didn't write it.
So from like the cloning vector, we'll begin from here, okay?
Yeah.
And the features of cloning vector. So
Yeah.
when we talk about the features of the cloning vector, so the most important thing that a cloning vector must have is the origin of replication. This is like a basic necessity, a basic requirement. So this is a sequence from where replication would begin, right? In any organism, the ORI is the sequence from where the replication begins. And whenever we try to link a piece of DNA into a vector, we require this ORI so that if you link it over there at the origin of replication, so whenever that, you can say sequence or that DNA or that plasmid will, you can say replicate, so as the vector will be replicating alongside. So the process of cloning or making multiple copies is done in this way, that you link the gene of interest to the ORI and thereby you get the product. Clear? Now moving forward, talking about the selectable marker. Now if you've seen the picture of the PBR, this thing, it's there in the book, the plasmid.
Yeah. Yeah.
So if you see over here, there are different sites conferring for different things. For example, there's Hint3 site, there's Eco R1 site, all of the different sites are there. And this particular plasmid, it has ampicillin resistance site, the AMPR, that is ampicillin resistance. Okay, this part. Whereas the second part is the tetracycline resistance. Understood? Now, the presence of selectable marker is one such thing that, for example, if you have taken a gene of interest and you have added it to the site, restriction site or anything of that sort. Okay, so then what will happen? Like, how would you know that the transformation has been done? So it will be done when that particular plasmid loses that resistance. So the selectable marker is that sequence, is that part of the vector which will help us to identify whether the vector has become a transformant, meaning has been transformed or has the gene, or a non-transformant that has no modification in the gene. So there are many genes in prokaryotes which will be coding for antibiotic resistance, right? Such as ampicillin, chloramphenicol, tetracycline, kanamycin. These are the different names of antibiotics. So what will happen if, for example, you put some sort of gene at the tetracycline resistance site? Now this plasmid is going to lose the resistance for tetracycline and it will be only resistant to ampicillin. So now you will know that if you put them in a Petri dish that contains tetracycline, it will die. But if you put it in a Petri dish containing ampicillin, it won't die. So now you will know that, okay, this has been transformed. It is living in ampicillin, but it is dying in tetracycline. Understood? So that is your selectable marker, which is helping you to understand that transformation has taken place or not. Did you get it?
Yes,
And
ma'am.
remember, amongst all the different types of bacteria, E. coli is the most studied bacteria. No matter whichever, prokaryotic research is done on E. coli and it's available very easily also. Now, the other type of selectable...
Can you find E. coli in the environment? Like just like that?
Yeah, it's
It's
there,
why.
it's in our
Okay.
gut also. Also, remember one more thing, like for example, now for this type of selectable marker wherein ampicillin resistance is concerned, so if you want to see whether or not the transformation has taken place, what you will do? You will put it in one medium, you'll put it in another medium, then you'll compare, right? Then you'll put it in a medium containing both. So you'll need to prepare media, then you have to perform like put it here, put it there, like that. You have to do all of these things, right? So what do you have to do then? That this will be a cumbersome process, isn't it? It's going to take your time also, and it's going to be tedious also. So what would be better if these vectors had something that would produce a color if recombinant or not? So color to we easily can see, and we don't have to do the plating or
Oh,
stuff.
yeah. Yeah.
So some enzymes such as beta galactosidase, they are coded by the LAC-Z gene. If you remember LAC-operon,
Yes,
this
ma'am.
is also used for selection process. How? This enzyme is going to utilize the substrate and it is going to produce a blue colored product. So this blue color production or white color colonies production will help us to understand which one is a transformant, which one is clear.
Yes,
So
ma'am.
selectable marker is clear to you.
Yes, ma'am.
Then there must be cloning sites in the vector. Now, cloning site is where the part of the vector where the foreign gene is linked at. Now, normally we would think that a vector should have a lot of cloning sites, but no. An ideal vector is the one that has the less number of cloning sites, like the perfect
Because
one.
they want to do the process properly. That's why it has limited
Yeah.
number. Yeah.
And if there is more than one cloning site, then obviously it's going to be complicated. It's going to become hodgepodge. That is why. The size of the vector should be small because large molecules, when you'll go for the purification, they'll break down. They'll break. And also it is going to be very difficult for us to transfer it into the host. That is why the size should be like smaller. Then high copy number. Copy number means the ability that it has to replicate and increase the number. That is its copy number. Understood? So high copy number must be. Now, examples of the vectors that are commonly used in the RDT, the first one, the most important one, is the plasmid. Now, you know that the plasmid, they are extra chromosomal DNA that are circular, right? Other than the genomic DNA, there is a plasmid DNA. It is autonomous and it is self-replicating. It is found in bacteria and it is found in some yeast. That's it. Now they have a lot of, you can say, antibiotic resistance property. Now see, from this particular diagram, this is your artificially made plasmid, PBR 320. Okay. So what you have to do is that this diagram gets asked as it is. They'll remove the labelings, ask you to label it. They'll ask you to draw it. Okay. And if not, they'll put it in the MCQ part and ask you BAM H1 confers which resistance. PVU site confers which resistance. Understood? Like that. Like if you think about BAM H1, SAL1, both of them are at the tetracycline resistance area.
You have
Right?
to write TET and then R, right? You don't have to write the whole name. Or
While you're drawing the diagram, you write TET to the power R. That means tetracycline resistance. They'll ask you in, like they'll give you this diagram, they'll ask you in the form of MCQs also, that if you are cloning any piece of DNA at BAM H1 site, so which of the resistance is going to be lost? So you should know that what all things are present at tetracycline resistance and what is present at the ampicillin. So while you're taking down your notes, you roughly make this so that you will remember it. Trust me, this can come in, to draw the diagram, to label it, or it can definitely going to come in the MCQ. Clear?
Yes, ma'am.
Now in this particular diagram, the HINT3, the ECORI1, then BAMH1, SAL1, PV1, all of these are the restriction sites. Here, ORI is the region, origin of replication, and ROP, it is the part of this wherein it would be coding for the protein involved in replication of plasmid. So ROP is replication of plasmid. Okay. Now the difference between plasmid DNA.
Okay, never mind. No, I'll tell anyways. You know what is ROP in Oman?
What is it?
Royal Oman Police.
That's
Yes, ma'am.
okay. We're talking about the differences between the plasmid DNA and chromosomal DNA, meaning how are they different from each other, whether or not they are different. So chromosomal DNA can be present like in a linear format also as well as a circular format. But the plasmid are always circular. Both of them will be double-stranded. They'll not be single-stranded. Now plasmid DNA has no packaging, so it is without histone protein, whereas the chromosomal DNA it is coated with histone protein. Then plasmid DNA does not carry any vital gene. It's not important for the necessarily cellular function, whereas the chromosomal DNA, they carry the vital genes. Now, the replication in the plasmid DNA, it is independent of the main gene. Meaning, it doesn't care. It replicates on its own, doesn't care about the cell. No one orders this. But the genome is ordered by the nucleus. Clear?
Ma'am, how is the plasmid? Can I see the first point again? It's double-stranded,
Two strands are there, no?
but
Because it's
it's
DNA.
okay.
The two circles you see, strands means it doesn't have to be straight. Two circles
Okay,
means
good.
double-stranded. Understood? It's circular in one circle, two circles. Now, selection using antibiotic resistance. The whole entire thing that I told you in language. So, the ligation of the foreign DNA, this is done at the restriction site where the antibiotic resistance is present. Now, if let's just say the ligation, you've done it at BAM H1 site. So, now the plasmid is going to lose the resistance to tetracycline because the foreign gene has been inserted at that time. Now, this particular plasmid is a transformant. It has been transformed because now it contains its own, no, it contains a foreign DNA. Transformant can be recognized from the non-transformant by plating on the tetracycline containing medium. Like you'll put it in ampicillin, you'll put it in tetracycline, you'll put it in a medium containing both. Now, in this case, one antibiotic resistant gene helps in selecting the transformant from the non-transformant. Why? Because of the insertional inactivity, this phenomena wherein they are losing the resistance because of the insertion of gene. So you call it as insertional inactivation.
So they have to select one, right? Any one.
You'll have to select transformant.
Okay.
I mean, that is what, like, as far as
Yeah.
the research is concerned, you are concerned only with the transformant. You don't need the non-transformant. It will be useless. Understood? Now, the disadvantage of this particular process is that it is cumbersome. Why? Because you have to constantly plate it here and there and there. So, another type of selection is the blue-white selection, which is also known as the chromogenic selection. Chromogenic means color-producing selection.
Would you
So,
write it?
it's there. Everything is there. So, in this type, the selection of recombinant from non-recombinant on their ability to produce a color in the presence of a chromogenic substrate, meaning a substance that produces color. So, in this, a recombinant DNA is inserted within the coding sequence of an enzyme, beta-galactosidase. Now, because of this, the beta-galactosidase gets inactivated. This phenomena again is insertional inactivation. Now, the presence of chromogenic substrate gives blue color if the plasmid is not. Now, see, if you have put the gene successfully, it will not give a blue color because beta-galactosidase will get inactivated because of insertional inactivation. So, the bacteria that produces a white colony is your transformant, whereas the bacteria that produces a blue color colony is a non-recombinant or non-trans. Clear?
Yes. Yes, ma'am.
Presence of inert result. Insert. Presence of insert results into the insertional inactivation, right? We have put that piece of DNA. So the beta galactosidase, because of that insertion, it is going to get inactivated. And then this beta galactosidase will not be able to use the chromogenic substrate and produce the color. So they will be the recombinant. The white ones will be recombinant. So I'll write it here. White are the recombinants, whereas blue are the non-recombinant. Non-recombinant means non-transformant. The meaning is clear.
Yes, ma'am. Yes, ma'am.
Other vectors like plasmid is one of the most widely used vector. Other than that, there are bacteriophages that like the viruses that eat bacteria. They are also used to deliver the gene of interest. Okay, because it has a very high copy
Yeah,
number.
it can insert. Yeah.
Then in the case of plants, specifically, there is TI plasmid. That is, TI stands for tumor-inducing plasmid, which is taken out from Agrobacterium tumefaciens. This particular bacteria has this plasmid, that is TI. So, tumor-inducing plasmid, meaning this bacteria, when it will reach the plant, it will cause, like if let's just say a plant got a cut or some sort of wound, so through that this bacteria will enter, and these plasmid will induce the formation of a tumor in the plant. Tumor means
Same thing as humans. So the cells will like keep
like for example, you took a TI plasmid. TI plasmid has a nature of creating tumor, meaning mass of undifferentiated cell, meaning it divides a lot. So now you take a plasmid, you put your gene of interest into it. Now you take that plasmid, put it in the plant. It will divide and the gene will be sent to the plant.
Plants also have that, like in them. So they also
Plasmid
can.
is taken from bacteria. Plants don't have it. We are using
No,
this
no,
plasmid.
yeah, this plasmid, if we put it in the plants, then even the plants will get
Have a
it,
tumor.
right? Okay.
So TI plasmid normally in general
So how do
the
they get it? I mean, they don't have.
they don't have what?
I don't know. Like, I mean, they have cells, but then
What do they not have?
I don't know. Like, so the plasmid will go and then do the same thing what it's doing to the humans. It will go and attack the cells, and then the cells will become huge in number, and then like that. So even the plant can die like that. Okay.
Plant. There are so many plant diseases: fungal, bacterial, viral.
Yeah, fungal, bacterial, and all is fine, but
All
okay.
sorts of disease, like tumor in the plants also, in the root also. You see big cells, clusters.
Oh,
Tumors are also there in the plant. So the TI plasmid, it causes disease in plants, and it has been like you can disarm it, like you remove that part which causes disease, and you'll modify it, and then you can transfer the gene of interest into the plant. Then talking about animals, so the vectors that are used to clone genes in the case of animals, like retroviruses. Now you know that how they use the our cell only as their factory. These are RNA viruses. So now if you unarm them also, and then you put them in the animal, responsible for the same thing. So disarmed retroviruses can be used to deliver the foreign gene in animal cells. Then there is also shuttle vectors used in the plants. So three kinds or like these different kinds of vectors which replicate in both eukaryotic cells also and E. coli also. These are artificially made vectors. So they contain two types of origin of replication and selectable marker genes that they can, like replicate in the eukaryotes and the prokaryotes. Okay, so yeast episomal plasmid, this is, and modified TI plasmid. These are your shuttle vectors. hosts, which are created artificially in the lab. Understood? Any doubts?
No, ma'am.
Now remember one thing, when we are doing the recombinant DNA technology, we have to keep in mind that the cell is not ready to take things. We have to make the cell ready, make the host ready to take up the piece of DNA that you're transferring. So that is a whole entire thing of making the host competent. Competent means compatible to take up the foreign DNA. So there are various
What if
things
it
we
is
do
not
to a host.
compatible? What if it
Because
is not?
normal, that's why it is not. But we make it.
We make, okay.
We make the host cell competent so that it can take up the foreign thing. Because normally cells are resistant to it, isn't it? It's like, I'll not take anything from the outside. It's selectively permeable. So we make it that, meaning we create some sort of entry point for the vector. Got it? Okay. Now you note down. Tell me the last thing you've written.
Hey, 1 second. 1 second. I'll just get the book. Working of RE.
Which one? Acha. What is the last line?
From example onwards.
Okay.
Yeah. Ma'am, can you scroll down? 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 Can you scroll down?